Hello, welcome to Study Hub Pro. Today, we are diving deep into the absolute core of what makes life, well, life. We're talking about molecular genetics. So here's our game plan. We're going to follow this incredible journey of information, starting with a single gene that's all locked up inside DNA. We'll track it all the way to a finished, functional protein that's out there doing a job in the cell. We'll kick things off with the master plan, the central dogma, then we'll walk through transcription, translation, and finally, see how the protein actually gets built. Let's get right into it.
All right, first up, the central dogma. Now, I want you to think of a cell's DNA like this enormous, precious library of blueprints. Every single blueprint has the instructions for a different, very specific machine. So the big question is, how does the cell find the one correct blueprint it needs at just the right moment and use it to build something like an enzyme? Well, that whole process is what we call gene expression.
To put it really simply, when you hear that a gene is being expressed, it just means the cell is actively reading that gene's instructions to make something useful. It's all about turning that stored information into real action. This whole process follows a set of rules that we call the central dogma. It sounds kind of intimidating, but the idea is actually super straightforward. Your DNA is the master blueprint, right? It's kept super safe. You'd never take the one and only master blueprint to a messy construction site. No way. So the cell makes a quick, disposable working copy. That's RNA, and the process of making that copy is called transcription. Then that copy gets sent out to the cell's factory, the ribosome, where the instructions are read to build the final machine. That's the protein. And that process is called translation. So just remember this flow: DNA to RNA to protein. That's the heart of it all. So let's zoom in on that first big step, transcription. You know, before the factory can build anything, it needs a really clear, easy-to-read copy of the instructions. The cell can't risk damaging the original DNA blueprint, so it has to make a copy, and that's exactly what transcription is. At its core, transcription is just rewriting information. The cell is copying a gene's sequence from the language of DNA into the almost identical language of RNA. The copy it produces is called messenger RNA, or mRNA, for a pretty obvious reason. It carries the genetic message from the DNA out to the rest of the cell. Okay, but hang on a second. This brings up a huge problem, doesn't it? The DNA molecule is gigantic, with thousands and thousands of genes. How on earth does the cell's copying machine know which gene to copy? And maybe even more important, how does it know exactly where the gene starts? It solves this problem by using a whole team of key players. Of course, you have the DNA blueprint itself. Then you have the star of the show, an enzyme called RNA Polymerase. This is the machine that actually builds the RNA copy. But it needs help. It needs to find the starting line. That's where a special start here sign on the DNA called a promoter comes into play. And often, there are also helper proteins called transcription factors that act like guides, leading the polymerase to the exact right spot. And, fun fact, in bacteria, these guys often work together in a unit called an operon, which lets one switch control a whole group of related genes all at once. Pretty efficient. Now, especially in bacteria, and this is a key concept, so listen up. These promoter start signs have two really important sequences. We call them the -10 and -35 elements. Honestly, the best way to think about them is like landing lights on an airport runway. They tell the RNA polymerase not just where to land, but also which direction to face. By locking onto both of these spots, the enzyme is positioned perfectly to start copying in the right direction. This is absolutely critical for the whole process to work. Okay, so our machinery is all set up and ready to roll. Let's walk through the actual process of transcription now, which, like any good story, has a beginning, a middle, and an end. It all happens in three main stages. First is initiation. This is where all the players get into position and the DNA is unwound and opened up. Next is elongation. This is the main event, where the RNA copy is actually built one piece at a time. And finally, you have termination. This is where everything grinds to a halt and that brand new RNA molecule is released. Now, stopping the process correctly is just as important as starting it. In bacteria, there are two major ways to hit the brakes. The first way is called Rho-dependent, and it uses a special helper protein called Rho. You can picture Rho as this little motor that jumps onto the new RNA strand and just zips along it until it catches up to the polymerase and basically shoves it off the DNA. The other method is called Rho-independent, and it's a bit more elegant. The RNA molecule itself has a sequence that causes it to fold back on itself into this really tight little hairpin loop. This hairpin acts like a physical wedge that just pops the polymerase right off the DNA. So one method needs an outside helper, while the other uses a cool trick built right into the RNA sequence itself.
All right, we've got our messenger RNA copy. It's done its job, it's out of the nucleus, and it's arrived at the cellular factory floor. So, now what? How does the cell actually read this message and turn it into a protein? Well, that brings us to our second huge process, translation. Translation is where we switch languages completely. We're taking the information written in the RNA alphabet and we're translating it into the language of amino acids. Those are the building blocks of proteins. And this whole incredible feat of engineering happens at the ribosome.
So this is the million-dollar question for translation, right? How does this amazing machine, the ribosome, look at this long string of RNA letters and make any sense of it? How does it crack the code?
Well, here's the secret. It doesn't read one letter at a time. It reads the message in 3-letter words, and we call these little genetic words codons. Each group of 3 letters is a specific instruction, usually telling the ribosome which amino acid to add next. Here's a look at the genetic code table. You can see how different 3-letter codons correspond to different amino acids. Now, if there's one codon you absolutely positively have to remember, it's AUG. Not only does it code for an amino acid called methionine, but it's also the universal start signal. It tells the ribosome, begin building the protein right here. And just like there's a start signal, there are also stop signals, codons like UAA that yell, all right, you're done, the protein is finished. Okay, we've got our instructions, we know the language, we know how to start and stop. So let's check out the final assembly line where this protein is actually put together piece by piece. And what do you know? Just like transcription, translation happens in 3 stages. Surprise, surprise. First, you have initiation, where the 2 halves of the ribosome come together on the mRNA right at that AUG start codon. Then comes elongation. This is where the ribosome chugs along the mRNA, reading one codon at a time. And for each codon it reads, another molecule, called a transfer RNA or tRNA, acts like a delivery truck, bringing in the one correct amino acid to be added to the chain. And finally, you have termination. The ribosome hits a stop codon, everything falls apart, and the newly built protein is released. But the story doesn't always end right there. Sometimes that brand new protein is like a car that's just rolled off the assembly line. It works, but it might need a paint job, or maybe some final parts need to be bolted on. We call this post-translational processing. It's just a series of final tweaks, folding, modifying, or even cutting the protein to make it fully functional. So let's just take a step back and appreciate this beautiful, elegant 2-step dance from DNA to protein, transcription and translation. It's not just some obscure pathway. It is the fundamental operating system for every living thing we know of. It is how life turns a static blueprint into dynamic living machinery. And that leaves us with one last fascinating question to chew on. We've seen how a protein is built, but think about it. A cell doesn't need every single protein working all the time. That would be chaos and a huge waste of energy. So how does the cell control when a gene gets turned on or off? Well, that whole world of control, known as gene regulation, is a fantastic story for another time. See you in the next video.